Hi, everybody. We're going to walk through most of the steps that you need to do for the protein structure project. The first thing you should notice about this is the primary structure of the polypeptides is listed right up here. So again, if you haven't noticed that yet, this is the primary structure. And you can see individual amino acid names here. Each letter represents one amino acid. And so C is cysteine, T is threonine, T is threonine, F is phenylalanine, et cetera, et cetera. If you need to identify a particular amino acid, just look up an amino acid chart as has been on the problem set. So this is where you're going to find that. You can copy, say, 10 amino acids, say D, E, I, F, R, S, T, D, T, L, Y, L. Copy 10. And then I'm going to ask you to come up with the properties of each of those. So you'll have to look on the chart. Hey, D and E, aspartic acid and glutamic acid, are both acidic or negatively charged. And so you'll mark that. The way I was doing it on the problem set is just by labeling it as an A for acidic. Isoleucine is nonpolar, and so you could just classify that as an N. And so I want you to just do a quick analysis of of one stretch of primary structure. So if I was going to do that, DEIF firstly, I'm going to probably get this wrong, but let's say that I was doing this here. I should screen capture it, but this is just uh, just trying this out. So let's see what happens here. That was probably close enough. DT. All right. What I would then do is describe the properties, and if you want to do it in abbreviated context, that would be fine. So if I wanted to do just strictly acidic, acidic, nonpolar, nonpolar, uh, basic, etc., you could do that for this particular requirement. Okay, next requirement, secondary structures. So going back to this protein, we can notice first in quaternary structure, we see three different polypeptides, each color-coded. They happen to be identical. And if you're wondering, how do I know that? Well, this is the polypeptide, spike glycoprotein, and it turns out there's three of them. And if I switch from one to the next one, you'll notice the primary structure is unchanged. And same thing for the third one. The third one just happens to be, if I look, I might be able to figure out which is which by seeing an amino acid get highlighted. Um, yeah, this is the purple one. So the next structures are the secondary structures. So what I'll want to do is zoom in, and I'm going to have to make some changes out here. So I'm going to collapse most of these just because they're sort of, we don't need all that complexity. The only one you want is components. And then to the right of polymer, this is going to be important, click the three dots. And what we're going to do is add a representation. And so right now we're in cartoon, and we could change this to something else like ball and stick if we wanted, click update for it to happen. I tend to think that this is too complex for us, so I like to keep it on cartoon at least for now. But we want to have the hydrogen bonds shown and other bonding types. So I'm going to add another representation. All right, so this is the current one. I'm going to add one more by clicking add representation, and I'm going to add non-covalent interactions. Now, two will be shown if I open this up again. There they are. Both of them are there. You might not notice an immediate change, but if we zoom in, we'll see all these little dotted lines everywhere. So what we need to do for this portion of the project is to actually zoom in on one of these secondary structure elements and screen cap it and highlight the bonds that are forming it. And so if I just zoom in on any one of these, and let's say we're going to look at this alpha helix, oh, a little bit too close, this alpha helix right here. So I could screen cap it. I'm going to grab this. Hold on. I'm actually going to highlight one of these bonds. All right. And if you look in the lower right-hand corner, down here, you'll see it highlighted. You'll see it described as a hydrogen bond. And let's see. Sure, I'll do this. I'm going to capture this region like so. And that image I'm going to drop into this document. So for example, this will be, I'll just delete this section and I'll say alpha helix, and I'm going to drop in that image right there. And what I'll want to also do is highlight what kind of bond that was. And so I'll want to describe that here. And I may want to even put in the specifics of what's being bonded. Now, I want to show you this. It is a hydrogen bond because all secondary structural elements are based on hydrogen bonding. Now, you may get a little confused though, because at the bottom, if you look carefully, it says the two amino acids are ASN, asparagine, and the other one is cysteine. Now, what you might get confused by is cysteine is generally nonpolar. But remember, we're talking about backbones. This is backbone interaction. So it doesn't even matter what amino acid side chains we're talking about. The backbone of the cysteine is the same as the backbone of a lysine, is the same as the backbone of an arginine. So it doesn't matter if the R group is polar, nonpolar, positive, negative. It doesn't matter in this case for the purposes of this course. So when you see the cysteine and the asparagine listed there, it's not the actual side group. It's the O off of a C. It's a carbonyl group bonded to an amine group on the backbone. Okay, spending too much time on this one. So that's one. I would then go and do the same for a beta pleated sheet or another alpha helix if I didn't have a sheet. But I do have a sheet. So I'll zoom in on a sheet. This is a pretty good one. Right here, I'll zoom on in. And you'll notice that instead of the hydrogen bonds being kind of vertical in the tube of an alpha helix, these are horizontal between one backbone and the one adjacent to it. Again, it's not spiraling. It's not spiraling. It is anti-parallel in most cases, or sometimes parallel. So you get one strand going this way, another strand going this way, and hydrogen bonds going across. So I could highlight that hydrogen bond there. And if you look at the lower right, you'll see it listed as a hydrogen bond. And now I can just screen cap that particular beta pleated sheet. As I did before, I would just drop it into this location and then describe it over there. Let me pull that image, and there it goes. Okay, so that's the first two secondary structures. Next, we want to look at a hydrogen bond that's not in the secondary structure. We want to look at a hydrophobic group. We want to find a disulfide. Let me see how quickly I can do that for you because I'm taking too long on this video. All right, so next, we're going to look for hydrogen bonds, disulfide bonds, and a hydrophobic cluster. The easiest way to find a bond that forms tertiary or quaternary structure, meaning between alpha helices or between sheets or between a helix and a sheet, is to actually just look for polar amino acids. So if I was going to just sort of scan this, there are certain amino acids that you can look on the chart are polar. For example, there's a glutamine here. Glutamine represented by this Q, is a polar amino acid. So if I click on that, I can zoom in on all the way to the atomic sort of level. And here's that glutamine. And you can see this is the side chain coming off the backbone. So there's the backbone, there's the side chain. And you can see that it's actually forming two different hydrogen bonds. The amine side of the glutamine's side chain, R group, is bonding in this direction to a backbone element, but this oxygen on the side chain of glutamine is apparently bonding to a serine side chain over here. So this side chain is bonding to this side chain. So this is a tertiary level, tertiary type of structure interaction. So I'll just hover there, screen capture this. And to make it easier on myself, I'm going to screen cap with the designation of the amino acids, and I'll just pop that into here, and that will give me my hydrogen bond to describe. So pop that in here, there it is, and I can now just use that screen capture to say that this is between glutamine 288 and serine 569. And then I'd want to describe why. Why is this a hydrogen bond? By describing the properties of serine and glutamine. Okay, and you have a great deal of very specific information as to which atoms or groups are interacting. Now, if we want a hydrophobic cluster, it's a little bit different. So let me show you that. So I'll zoom back out. Actually, let's look for an ionic bond. This is not required, but if you want to find an ionic bond, don't look for just a polar side chain, look for a charged one. So like if we look for, oh, that's one right there. A lysine is charged. That tends to be positively charged. So if I click on that, here it is. Maybe we can find if it's forming a hydrogen bond. Let's see. This one appears to be on the outside, so it's not actually forming a hydrogen bond. Sorry, or an ionic bond, but maybe a different one. Let's see. I can find another one. Again, ionic bonds are a little bit harder to find because they're not the most common thing. They really are not. So again, if you're looking for an ionic bond, look very specifically for charged amino acids, and you may have better luck that way than I'm having. Okay, so let's look for a hydrophobic cluster, so a hydrophobic group. So hydrophobic is going to be nonpolars. Let's see what I got here. Oh, yeah, there's an ionic bond. It's not going to be labeled an ionic bond, unfortunately. That's kind of annoying. So ionic bonds are not labeled as such on this because of the strength of the bond is not truly like a salt bond. It's a little bit different. But if you look carefully at this, and I'm going to screen capture this, this bond between the R group of a lysine and the R group of an aspartic acid would qualify as, for our class, ionic, because lysine is positively charged, aspartic acid is negatively charged, and there is that bond. So if I wanted to include that as sort of my optional, my other, I could put that in here. So I'll put that right here. And again, if you screen cap it correctly, you've got the numbers and the identities of the amino acids to help you out there. Okay, hydrophobic cluster and disulfide bridge. Disulfide bridge is actually one of the easier ones to find because there's only one amino acid that makes disulfide bonds, and that's cysteines. So cysteines, here we go, are the only ones that can do this. So all you have to do is search for cysteines. And here's one, C for cysteine. If we zoom in, hey, it worked. So the great thing is cysteines have sulfhydryl groups, SHs, at the end of their side chain. So here's one cysteine. Here's the other cysteine, and you can see the two yellow balls are actually joined together by a covalent bond. So this right here, whoa, I lost it. Let's do that again. There it is. That is a disulfide bridge. And so I can just hover over that, screen cap this, or maybe even screen cap it without all of the sort of annoying like hovering. And either one, I can now drop into this document as a disulfide bridge. And I'll pop the second one in. So there's my disulfide bridge. And on a previous image, I'd gotten, let's see, CYS. Oh, and I didn't get the other one. But I'll be able to write down what those cysteines were. So cysteine, say 128, and its partner, which I could go back to my structure and find it. So there's 115 and 146. Okay, so I got it wrong. So cysteine 114 and 146. And again, we will have to specifically describe what's forming that bond and why. Okay, next, hydrophobic cluster. This video is taking too long. Hydrophobic cluster. The way to find these, you could do it manually, which is going to take be kind of annoying. But I'm going to show you a way of cheating this to make it a little bit easier. Instead of cartoon representation, let's switch it. Let's edit this and switch from cartoon to, let's see, Gaussian. Update that. So we'll see it from the surface. And then I think we can do this chain ID. Let's see. Residue property. So you'll have to do this slowly as I click through it. Hydrophobicity. Now, each of these colors means something in particular. Red means acidic, also known as negatively charged. Orange means basic. All right, so that's going to be positively charged. Anything that's sort of moderate is just going to be polar. So these sort of like light yellows, these are just polar amino acids, while greens are nonpolars. So if you want to find a hydrophobic cluster, just look for a green pocket somewhere. Green, green. All right. Tryptophan is very nonpolar, very hydrophobic. Tyrosine is moderately hydrophobic. And so I can just pick any of these spots. All right. I can just pick any of these spots and describe the cluster. So here I've got a phenylalanine 347. Maybe I'll write that down on paper. Tryptophan 410, also hydrophobic. Behind it, phenylalanine 348. Behind it, phenylalanine, oh, there it is again. Behind that, phenylalanine 351. Write those down and maybe capture this particular area. Okay, so I'll just capture that location. And then after dropping it in, I'll list the amino acids that form that particular cluster. So back in here, hydrophobic cluster. And again, to get that hydrophobic labeling, you'll have to follow what I just did on the video. And so here, I'll list the amino acids. And so there were some phenylalanines. There were, here we go. Let's get back to where it was. There is tryptophan 410, phenylalanine 347, tryptophan 410, phenylalanine, I don't know, 347, something like that. I would write those down and then transfer them in here. And again, describe, etc., why those are all bonding, all right, or being stuck together in one location. Okay. Overall, I think we've already covered that in that we've got, if I pull back and go back to cartoon, the colors used to show as the different chains. It doesn't anymore because I've messed this up by doing the hydrophobicity. But if I reload this, the default coloration is by polypeptide, and we can see 3 different polypeptides. And so you'd want to definitely describe that. And in my case, this was threefold symmetric. These 3 are identical. So this is a homotrimer is the technical term. So 3 things that are identical. So homo for the same, tri for trimer for the same, for 3 of the same. And you'd want to describe that here. So this is where I'd put the image of the whole protein. Okay, the last step here is going to deal with something that we haven't talked about yet. And we have generally talked about the idea of mutations in the DNA sequence or the gene sequence for a protein adjusting its structure. And that's what I want you to think about in the last part of this project. So we're going to go to a site called ClinVar. And this is going to be for anything that is a human protein. If you don't have a human protein, you're just going to have to do sort of a Google search for, like literally type in mutations in spike protein. You just type in the exact thing that you're talking about if it's not a human protein. If it's like a viral protein or a bacterial protein, just type that in and you'll get results. And if you need help, let me know. But let's say we're doing a human protein. I'm just going to use one, KRT1. This is keratin, one of the most common proteins in your body. It's the stuff that makes up your skin surface and your hair. And so this is going to be kind of overwhelming looking. And so this is a whole lot of different mutations that are listed here. Let me walk you through. This is where you get the information on the particular mutation. But what I want you to do is I'm going to ask you if this is your gene name, all right, go over to the left and click down here, missense. And this is going to indicate, this is going to be the type of mutation that changes one amino acid. Okay, that's what we're looking for, something that changes one amino acid. So in this case, we can look at this. This changes 1912, the alanine, sorry, whoops. This changes the adenine at nucleotide 1912 to a guanine in the gene sequence. The effect is it changes a threonine at amino acid 638 into an alanine. And so what I'm going to ask you to do is find a mutation in your protein and then identify why you think it would matter. And so just to give you an idea, if the mutation was, say, one of these amino acids, let's say it was this lysine right here. And I think this is the lysine. Let's find out. There it is. If this is the lysine, and this lysine, which is positively charged and is forming an ionic bond right here, actually, with an aspartic acid. If this was changed from a lysine to, I don't know, an alanine, what would that do to this part of the structure? Would it still have the same bonding ability? Would it still be able to form the ionic bond here? Would it be pushed somewhere else? So that's what I want you to predict. Look up a particular mutation in your gene, and then postulate, make a prediction as to how that might or might not change the structure of your protein. There is no perfect answer. You're going to be doing the best you can with just understanding that, in this case, over here, a lysine at 633 was changed to an arginine. Look at the properties of those amino acids and predict whether, in that structure, if you zoom into the structure, changing from a positively charged lysine to a positively charged arginine would have an effect. I kind of gave you a hint right there as to that one. There's also hints over here. If it says benign, it didn't really have an effect on the person. So that might give you a hint, but you don't have to necessarily agree with that. You could say something different. Okay, so I've gone way too long. So that hopefully covers most of the parts of the project that you need to know about. The first part's up here. You've already answered at least partway, but you can always revise your answers, but put those in there. All right, I'm going to stop talking. Have a good weekend, everybody. I will see you in class next week.